So let's talk about muscular dystrophies, what they are, and the different types of muscular dystrophies. So this video we're going to focus on dystrophinopathies, and essentially what are dystrophinopathies? Well, it's kind of in the name. Dystrophy means something bad, trophy means anything to do with growth or nourishment. What is it? Well, these are muscular issues such as muscle weakness and kind of lack of dependence on their own muscles that can be due to muscular dystrophy, or sometimes it can be due to neuropathy. But what distinguishes muscular dystrophy from neuropathy specifically is that neuropathy is when there's something wrong with the neuromuscular junction. Either the presynaptic or the postsynaptic cleft, there's something wrong with it. There's some kind of detriment to it. However, muscular dystrophy, the neuromuscular junction is usually fine. So a muscular dystrophy, a type of it would be dystrophinopathy. And again, it's in the name. Dystrophin is our protein. And opathy, it just means pathology of the dystrophin protein. So dystrophin is a protein that specifically stabilizes the sarcolemma. Types of dystrophinopathy are Duchenne and Becker. And Duchenne, if you remember, D for dangerous, it's the one which is very, very severe, the severe form of dystrophinopathy. Becker is the milder form, like Beck is mild. Okay, with Duchenne, it's the basis of it is a deleted dystrophin gene. Okay, this is very dangerous. It's X-linked, and it's often due to a frame shift or a nonsense mutation, whereas Becker is an X-linked disease due to a non-frame shift mutation. Like I said, Duchenne is more severe and symptoms appear very early on, by age 5 or even earlier, and Becker, it's less severe and the symptoms can appear in adolescence or even early adulthood. Now, that answers the what of it. Now, going further on about what it is, the dystrophin gene is a huge gene. Somewhere I read that it is the largest human gene that's vulnerable to mutation. But I know for a fact that it is the largest protein encoding gene. Now, it's so large, meaning that it actually has 79 exons and 2 million base pairs, whereas normal genes can have as little as only 10 exons and 50,000 base pairs. So this is a truly massive gene. What's the issue with large protein encoding genes? Mistakes can be made, especially during meiosis. Now, if you remember MM and DD, mistakes can be made during meiosis due to deletion, duplication, or point mutation. Now, this gene is found particularly on the X chromosome and on the short arm. This is represented by XP, and it's at the 21.2 location. So this gene is XP, so X chromosome, X-linked, and on the short arm, so P. Okay, so moving on, because it's X-linked, it's more common in males. So if we just look at the picture, this is a male genetic picture. He's got X and Y chromosome. Because he only has one X chromosome, if the gene is defective, that will manifest. Okay, so only one X chromosome, meaning that only one defective gene is required for it to be shown on the phenotype. Now, whereas in females, because they have two X chromosomes and either one of them can be inactivated due to the process of lionization, remember X chromosome inactivation, that means that one dystrophin gene on the one X chromosome can be functional, but the other one might be defective, and now it all depends on which ones are inactivated. Since girls have another functional one, they might have another functional one, they can appear to be asymptomatic. Or if it's the defective one, they can be manifesting carriers as well. Okay, now quickly moving on, where is this dystrophin even found? Well, we talked about the gene, let's talk about the protein. Dystrophin protein stabilizes the sarcolemma. So remember, stabilizes sarcolemma. The way that it's actually set out is that it anchors the actin. So if this is in the insides of the cell, it anchors the intracellular actin, which makes up your cytoskeleton of the cell. So it anchors your intracellular cytoskeletal actin to your dystrophin-associated protein complex. Again, so it's anchoring your actin to this complex that's within the bilayer. So actin, dystrophin is connecting your actin to your DAPC. Your DAPC is your dystrophin-associated protein complex. So your dystrophin is once again is anchoring your actin to your DAPC, which then anchors to your extracellular matrix. And this way, you can think of dystrophin as the wooden beam that the roof of a house is connected to. Okay, it's connected. It helps anchor the muscle fibers, and it connects your intracellular actin to the DAPC. Okay. Now, this clinical picture is very important, and it's often used in diagnosis. These kids have often a waddling gait, and because basically they can't depend specifically on their leg muscles. They have weak leg muscles and weak thighs, and due to this, if they fall down, these kids to get back up, will use their upper extremities. So they will use their arms to push themselves back up. And that's known as a Gower's sign, Now, in terms of also the waddling gait, they also have calf pseudohypertrophy. Now, something that I found really interesting is, why is it called pseudohypertrophy and not hypertrophy? Because this, basically, the muscle that is there, and now this is an example of Darwin's, was it Darwin? The use it or lose it theory, basically. And essentially, if you're using the muscle, fine. But if this gets degraded, then that muscle is not going to be used as often anymore. And that's known as myonecrosis. So basically, this is called calf pseudohypertrophy because the muscle is not replaced by muscle. Again, it's replaced by adipose tissue and some other fibrin or something. So it's adipose tissue, it's fat. That's why it's pseudo, like fake hypertrophy. So also, that's it really. Myonecrosis is happening. If myonecrosis is happening, muscles are breaking down and they're dying. Creatine kinase is going to escape through this broken structure. High levels of serum creatine kinase is to be expected in diagnosis. You need to check for mutations. So the tests you need to do are DNA test and Western blotting. You can also check the muscle for dystrophin stains. Treatment will be glucocorticoids, but these will not actually treat it. There's no cure. It will kind of just slow down the progression of the muscular dystrophy.